So we're talking today with dkNET, the team at dkNET, which is composed primarily of Jeff Greth, PhD, Koei Lin, PhD, and Marianne Martone, PhD. And together they've built a really sort of a deep collection of tools and resources that are available to the public research, that really through any T1D researcher and others who are interested in using them to create new ways to do science, as well as they're going to be unveiling a hypothesis center, which is really going to be exciting. And we will be talking even more about that on Thursday when we unveil a challenge we've built together, and will require the use really of all these tools and the hypothesis center. So it's really something to look forward to. Welcome, Marianne, and you're here today to talk about FAIR data and what it really means, why it's important. And thank you all so much for joining us. So do you want to take it away and start letting us know what is it that's so important about FAIR data?
Well, I was going to share I have some slides, but I can't seem to share them. Is that possible to allow me to do that?
Yep, everyone can share. It's.
Oh, now I can. Great.
Okay.
Okay. So thank you very much, Monica, for inviting us to speak about the work that we're doing at dkNET. And I'm entitling this presentation, The FAIR Data Principles, dkNET and You, which is everything that you will need to know about the FAIR data principles. And I'm going to tell you why in a moment that you would like to know about them. As Monica indicated, I'm one of the co-PIs of dkNET along with Dr. Greta. I'm also a professor emerita at UCSD, and I am a founder of SciCrunch, which is a tech startup out of UCSD, and it develops tools and services supporting reproducible research, including research resource identifiers, which I will talk about today. So I wanted to fully disclose that. So the learning objectives of this bootcamp are the following. First of all, I'm going to go over the new NIH data management and sharing policy. Normally, when I give talks like this, I have to promote open science and tell people why you should really be sharing your data. But thankfully, now NIH has done that for me, and you're going to have to start sharing your data in 2023. So I'm going to skip over the open science parts of it. The second part is going to be data management for sharing, right? What is it that you need to do to manage your data so that it is ready to be shared in accordance with the NIH policies? I'd like you to develop some familiarity with the FAIR data principles, what they are and how they apply to data management and sharing. And then I'll talk a little bit about how you can incorporate the FAIR data principles in your work. And of course, since this is a dkNET webinar, understand how dkNET itself can help you in your planning over the data lifecycle. So first of all, I just want to give a brief introduction to dkNET, just to give you the context. dkNET is an information portal that was established by a grant by the NIDDK, the National Institute of Diabetes, Digestive, and Kidney Diseases. And it was designed to make it easier for researchers, students of all career stages, to find research resources. By that, we mean materials, tools, funding opportunities, literature, news, and projects that advance the mission of the NIDDK. So NIDDK is a one-stop shop for being able to find these, but we also provide tools and services to help researchers navigate the many different policies that are coming out from NIH around rigor and reproducibility, sharing data, research resource identifiers, and the FAIR data principles. So if you look at the different portions of the dkNET portal, you will see that we offer both discovery services that allow you to go ahead and find information about what antibodies, cell lines, and things you should use. But we also provide these types of supporting services to make it easier for you to comply with the mandates and also just practices that are associated with sound science. And Monica's already indicated that the Hypothesis Center is a gateway to powerful bioinformatics tools, and we'll be hearing about that separately on Thursday. So there's basically the five areas in the portal: resource reports, authentication reports, FAIR data services, discovery portal, Hypothesis Center. We will have other boot camps where we'll go over these different portals into dkNET. But really, if one puts them all together, you see that dkNET can help researchers across the life cycle of an experiment. Helps you do background research to try to find out what's out there. allows you to construct hypotheses using bioinformatics tools like the Hypothesis Center, plan experiments, find the tools that you need to be able to do your experiment, help you understand how to collect and analyze data, and then finally make those results available and publish your data in a fair manner. So we really try to support the researchers across the entire life cycle of what it is that they are planning on doing. So for today, I'm really going to focus on this section of the portal here. It's called Authentication Reports and Fair Data. And of the two, it's really the fair data resources that I'm going to focus on. So I mentioned that NIH was soon going to be enacting requirements for all researchers to manage and share their data. And that target date is January 25th, 2023. And on that date, any grant application is going to have to include a plan for how you are going to manage your data inside your laboratory during the lifetime of that project and how you are planning on sharing it.
Can I just jump in here 1 second and highlight something that really struck me is this is not something that you can just say, oh, I'm going to start this on January 1st or anything like that, correct? This
Exactly.
is really something people have to get into the water with right now.
Exactly. That's why we're here now. We just finished a big workshop at the National Academy of Sciences that the NIH commissioned ahead of this policy well over a year and a half, because they recognize that the biomedical workforce is going to have to prepare to both implement and understand what is going to be required of them. And there's two things that are in here that are interesting, right? NIH knows that not all data can be openly shared. So there's human subject data, right? There's things where it cannot be shared. And the overarching principle is everything needs to be as open as possible and as closed as necessary. But you are going to have to justify for any data set why it is that you cannot share it. And that has to be approved by the NIH program officials. And they also have an interesting line in there, which I thought was very good. All data must be managed, because whether or not you can openly share it, it can all be managed, but most data should also be shared. So recognize that their policy is not just a data sharing plan, it's not just a data management plan, but it's a data management plan plus a data sharing plan, which they are calling DMS. And that's how I'm going to use this here. So very, very important. This is going to impact everybody who receives funding from the NIH, which is the largest funder of biomedical research in the world. So pretty much everybody. So as I mentioned before, I normally start my talks with why data sharing is good for everybody. But since we have that mandate, it really doesn't matter whether you think it's good for you or not, right? This is coming. However, when we did the workshop that I mentioned from the National Academy of Science, there was just a palpable sense amongst the people who are participating at all levels that this should not be seen as a burden, but this should be seen as a benefit. And who are the beneficiaries of making data available and managing data well? Well, number one, science. We really believe that science will be advanced through transparency, better reproducibility, reduced waste by understanding that experiments have been tried before, and of course, in the age of machine learning and artificial intelligence, really being able to drive discovery. The AI ML machine wants data, it'll eat it up. And so the more data it has, the better these algorithms can become. So first and foremost, they're doing this because, of course, their mandate is to have an impact on human disease. And I think everybody at the Sugar Science can appreciate that. But we also wanted to point out that there are actual benefits to you as a researcher. And we basically divide these both into you right now and you in the future. So one of the most likely people to benefit from data management and sharing through stable archives, as we'll talk about, is you a year from now when the journal comes back and says, I'm sorry, right, you need to share the data that underlies this figure, and you actually can find it. So the benefits to you is that this emphasis on data management is something that benefits you in your lab, it benefits your colleagues, it benefits your PI, and the things that you need to do to manage your data well are exactly the same things that you need to do to share it well, right? So these two things are very closely aligned. And I don't have time to talk about this today, but there are actually new credit systems that are developing so that you can get credit for sharing your data and publishing your data the same way that you can get credit for publishing a paper. Okay, so data management and sharing is good for science, for you now and in the future, and of course, your colleagues. And those colleagues may be people that you want to collaborate with. They may be your principal investigator, other people in your lab, and of course, right, they're the wider scientific community. So basically, we feel, that's why there's little happy faces here, that even though one can look at this as a burden, it's really an opportunity. We don't often get to live through such a fundamental transformation of the way biomedicine is done. And everybody in the biomedical enterprise is being asked to participate in this, to define what works for them, what they need to do at their institutions and elsewhere. So it's a really exciting opportunity. But why does one really need this? I think, why did NIH focus on data management and sharing both at the same time? And this is a video that was created in 2012. I know Jeff has seen this many times, but I think that this is one of the best illustrations of what the current problem is with the way that we handle data in our laboratory, even if we want to share it. So if you'll just bear with me, I'm going to play this video that everybody can hear, and I think it will set the stage very, very well.
Hello. My name is Dr. Judy Benign. I'm an oncologist at NYU School of Medicine.
Hello, Dr. Judy Benign.
I read your article on B-cell function. I think that I could use the data for my work on pancreatic cancer.
I am not an oncologist.
I know, but I think I could use the data for my work on pancreatic cancer. Do you have the data?
Everything you need to know is in the article.
No. What I need is the data. Will you share your data?
I am not sure that will be possible.
But your work is in PubMed Central and was funded by NIH.
That is true.
And it was published in Science, which requires that you share your data.
I did publish in Science.
Then I am requesting your data. Can I have a copy of your data?
I am not sure where my data is.
But surely you saved your data.
I did. I saved it on a USB drive.
Where is the USB drive?
It is in a box. It is in a box at home. I just moved.
But can I use your data?
There are many boxes. So many boxes. I forgot to label the boxes.
Hello again. Thank you for sending me a copy of your data on a USB drive. I received the envelope yesterday.
You are welcome, but I will need that back when you are finished. That is my only copy.
I did have a question.
What is your question? You might find the answer in my article.
No. I received the data, but when I opened it up, it was in hexadecimal.
Yes, that is right.
I cannot read hexadecimal.
You asked for my data and I gave it to you. I have done what you asked.
But is there a way to read the hexadecimal?
You will need the program that created the hexadecimal file.
Yes, I will. What is the name of the program?
Cytosynth.
I do not know this program.
It was a very good program. The company that made the program went bankrupt in 2007.
Do you have a copy of the program?
I do not use this program anymore because the company that made it went bankrupt. Maybe you can buy a copy on eBay.
I have good news.
You again.
I talked to my colleague. She knew a person with a copy of the software.
Then why do you need me? Everything you need to know about the data is in the article.
I opened the data and I could not understand it.
If you have the program, you will find it is clear.
Well, I noticed that you called your data fields SAM. Is that an abbreviation?
Yes, it is an abbreviation of my co-author's name. His name is Samuel Lee. We call him Sam.
I see. And what is the content of the field called SAM1?
Ah yes, SAM1 is the level of CXCR4 expression.
And what is the content of the field called SAM2?
That is logical if you think about it.
What is the content of the field called SAM2?
I don't remember.
What about SAM3? Is there a guide to the data anywhere?
Yes, of course. It is the article that is published in Science.
The article does not tell me what the field names mean. Is there any record of what these field names mean?
Yes. My co-author knows what the content of SAM2 is, and SAM3. And SAM4.
Can I talk to your co-author?
I'm not sure.
I would very much like to talk to your co-author.
Well, he was a graduate student. He went back to China 2 years ago.
Can I have his contact information?
He is in China. His name is Sam Lee.
I think I cannot use your data.
You could check the article to see if what you need is there.
Please stop talking now.
So what you're really seeing is that there's a failure here of the way that the data is managed in the lab. And that even though this here is data management on request, even if they request it, they can't pull it together. A study by John Borghi and others on looking at data management practices in the laboratory, and they created this very nice matrix. And it basically says, well, you can go from sort of ad hoc, right? When it comes to my data, I have a way of doing things, but no standard or documented plans. When I organize my data, I don't really have a consistent approach for it organized. So sometimes it takes time to find things. And then saving and backing up your data. I decide what data is important while I'm working on it, and I typically save it to a single location. In this case, it was a USB drive that turns out belonged to somebody who moved back to China and nobody could access them, right? So basically, this is the state of data management in most laboratories now. It is a somewhat ad hoc approach. Some people in the lab are better at it than others, but nobody thinks about managing data for the future and also for their colleagues. They think about managing it for themselves. So what is the desirable outcome, right? The desirable way to do this is what they call optimized for reuse. So I've created plans for managing my data that are designed to streamline its future use by myself or others.
Marianne,
Yes?
could I just interrupt you for one moment and ask you to share your screen so we can see the slide?
Oh, it's not shared?
No.
Oh, I think when I went back on, I didn't share the screen. No wonder it, I thought that it had shared the screen. So no wonder you didn't see the video because I wasn't sharing.
Oh, well.
So
Okay.
it looked
Oh,
like it would do.
This is part of the boot camp grueling.
Sorry
days.
about that.
That's
Okay.
fine.
Yeah, no, I wish you'd told me that before. So this is just what I was showing, is that there's this, this matrix that they have created. So I organize my data so that others can navigate and understand it and use it without me being present, right? And I save my data in a manner and location that is designed to maximize opportunities for reuse by myself and others. So this is the optimized for reuse. This is the ad hoc. And most researchers are here. A few of them are sort of in between. But what NIH and others really want you to get to is this point out here, right? That you know in advance, before you even do your study, what you're going to do with this data. You know what the endpoints are. You know how you're going to store it, how you're going to annotate it, how you're going to make sure that it is useful for others. But you might ask yourself, well, how do I manage my data so that it is going to be likely to be reused by others? And that's where the fair data principles come in. So if you read the NIH data policy, you see that it says, NIH encourages data management and data practices consistent with the fair data principles. And NIH strongly encourages the use of established repositories to the extent possible for preserving and sharing scientific data. So I put both of these together because in my view, they go hand in hand. When one thinks about FAIR, you think about publishing your data in an archive, which really helps you achieve FAIR. So let's look a little bit closer at the FAIR data principles. So the FAIR data principles were published in an article in 2016 by members of an organization called Force 11, the Future of Research Communications and E-Scholarship. And the acronym FAIR stands for Findable, Accessible, Interoperable, and Reusable. If you want your data to be able to be reused again, first people have to find it, then they have to be able to access it, They need to be able to combine it together with other things, understand what's in it, and then they need to have special characteristics of it to make sure that they can reuse it. And these principles, this acronym, really came out of people such as Jeff and myself and others around the world who were looking at public data and commenting that, in fact, even if you could find it, you couldn't understand it. And even if you could understand it, it might be in a form where you couldn't reuse it. And so this represented a lot of people working across a lot of different domains, looking at what the problems were of public data on the web. So let's look at the FAIR data principles in a little bit more detail. I always like to say that the acronym FAIR is like mom and apple pie. Who is against these, right? Who doesn't want their data to be fair? And a lot of people say, yes, I want it to be findable, accessible, interoperable, and reusable, but they don't bother to look at the FAIR paper itself, where they actually make some very specific recommendations about how you do that. And a lot of these are fairly cryptic, and one may not know what they mean. For example, data and that meta in front refers to metadata are assigned a globally unique and persistent identifier. What the heck does that mean? They're described by rich metadata. They have licenses, they use fair vocabularies, right? All of these things are what go together into making something fair. And if I were a researcher who was looking at the FAIR data principles because I bothered to read this, I would be extremely confused. So we're going to go into them in a little bit more detail and the parts that apply to you. But first, this is to remind me to make sure that I define metadata so that you know what it is. So metadata basically are data about data, for example, file format, file size. And descriptive metadata describes a data set for purposes such as discovery, identification, and use. So the study organism you use, the condition studies, those are all considered descriptive metadata. And when I talk about FAIR and I talk about metadata, that's the type of thing that I'm talking about, because as you'll see, metadata are extremely important to achieving FAIR. Okay. So first of all, as I mentioned, you could look at all 15 of those and your eyes might go, I have no idea what half of these means. And the good thing is you don't have to as a researcher. The majority of FAIR is actually falls on those who are running public repositories or even private repositories and archives who are specialized for handling your data. So you submit your data to them, much like you submit an article to a journal, and they stewardship for that then transfers over to them. So they're now responsible for maintaining your data, and they're responsible for doing a lot of things in FAIR. So we'll talk about repositories hand in hand with the FAIR data principles all the time, which is why I wanted to introduce this. But what do you as a researcher have to pay attention to if you really want to share FAIR? Well, not a surprise, good data management in your laboratory. We've already covered that. You're going to have to pay attention to the metadata that you collect about your data and that you make available. You've got to think about what it is ahead of time that you're going to need to do to achieve FAIR before you share. So you need to understand a little bit about what's expected of you. One of those things is that you're going to have to understand what standards might be available in your particular domain for collecting data or annotating data, right? So you need to know about those, and we're going to go through all of this. And finally, you need to submit it to a repository, right? You don't post them on your website, you don't publish them in supplemental material. You submit it to a repository who is responsible for all the rest of the parts of FAIR, okay? So if you only learn a few things from my lecture, that is a big one, right? That the end goal of a data management and sharing plan, as per NIH's own words, is that you're going to submit it to a repository who is going to become a responsible steward for your data.
I might just add right there that dkNET is such a repository, correct?
Actually, no. The dkNET itself doesn't host data except in extreme circumstances, but dkNET can put you in touch with the repositories that you will use, and we are going to go over that.
That's what I meant. Like if you need to source a repository, dkNET can connect you, basically.
Exactly, and we're going to go through how dkNET does that in just a moment. So I want to go through this findable principle in a little bit more detail, specifically the first two, which is that data, metadata, again, I'm using them interchangeably, are assigned a globally unique and persistent identifier, and data are described with rich metadata. Because Even though, again, the repository is responsible for assigning this persistent identifier, it's really important that researchers start to understand how important identifiers are to achieving FAIR. In fact, they're so important that in the data sharing policy of NIH, they say researchers are going to be asked to describe how the scientific data will be findable and identifiable, i.e. via a persistent, unique identifier and other standard indexing tools. So this is really integral to FAIR. And if I were a researcher, again, just being introduced to this, my first reaction would be like, huh? Of all the things that I need to achieve for FAIR, why is this the first one? Why is this the most important one? And it basically says it in some of the sites that attempt to explain FAIR. It's arguably most important because it will be hard to achieve any other aspects of FAIR without these globally unique and persistent identifier, or PIDs, as we call them, PIDs. Hence, compliance with that will already take you a long way towards publishing FAIR data. So what is a globally unique identifier? It means that in the digital world, it is unique. So if there's only one object that is identified by that identifier, and it is never reused. So it points to that object for all time. It's also stable in that it continues to point to that object, even if that object moves addresses. So for example, if I myself, Marianne Martone, had her own ID, which I do, my ORCID, and I moved from UCSD to someplace else, I'm still Marianne Martone, I just happen to be in another address. And so it really doesn't matter where I go, I still can receive my email, right, so to speak. So they're really very important, and they are both unique and they are stable. But one of the things that's really important to know about these is that they're only stable because human beings are standing behind them and making sure that they're stable. So there's nothing magic about the computer. A lot of researchers think, so I just need to slap an ID on it. It's like, no, you don't, because you need some organization like these data repositories to stand behind them. And as we'll talk a little bit about, they're resolvable. That is, these are really designed to work on the internet. I can plug them into a web browser and I can be taken to that object no matter where I am. So I can find Marianne Martone on the web, independent of whether she's moved from UCSD to someplace else, because it knows me as an individual. So these persistent identifiers, or PIDs, are so important that there's actually a scientific conference that is devoted to them called PIDapalooza, and it meets every year to really discuss these PIDs. So that's why I want to spend a little bit of time on them. So here's some examples of persistent identifiers. You're probably all familiar with the digital object identifier, or DOI. This was developed by the publishing industry, and so you see them on all scientific articles. If you look at the metadata associated with an article, you'll see a PubMed ID, you'll see a PubMed Central ID, and you'll see a DOI. This one is issued by the publishers. These are issued by PubMed Central, but they're all cross-mapped to say these are all the same object. But I already mentioned the ORCID, which is the digital object identifier, if you will, for individual people. And if you've published recently and submitted an article for publication, you're asked for your ORCID, so that it uniquely identifies you. And we're also going to talk a little bit about these research resource identifiers. These are the persistent identifiers for research resources like antibodies, organisms, and others. So there's many types of these. And as I mentioned, they are issued by special organizations who issue them. This is not something that you do yourself. So here's some PIDs in action. This is a project called SPARK, which is mapping the autonomic nervous system and its interaction with end organs. And this is a data set that's on a human islet microvasculature analysis. So this is a data set, and it's got a DOI associated with it. And if I click on this link, it will take me to the landing page of that data set. So it'll come over here and it'll say, here it is. Here's the entire data set. You can see the full picture. You can see, who created it, so on and so forth. So that's how a resolvable identifier works. And if you can see up here and your eyes are very good, you'll see that the URL is actually not that DOI, right? This URL references the location of that data set, like UCSD for Marianne Martone, but the DOI doesn't care. It says, okay, this happens to be found here. It could be found someplace else as well. Are you seeing the slides again? Because it's always bad when you go out of them. Are you seeing them?
Yes, it's perfect.
Okay.
Yep.
Okay, great. Okay.
Good.
So my lesson for this is that, again, you cannot assign a PIDs for yourself. The way that you get them is you submit to a trusted repository that provides these. And as Monica indicated, dkNET is a place which can help you find that repository for your data. The second most important principle of FAIR, which is the reason why it's number 2, is that data are described with rich metadata. And you might say to yourself, well, what exactly is rich metadata? Actually, the FAIR data principles gives a lot of leeway to individual scientific communities to be able to define them for themselves. But really, rich metadata has come to refer to the fact that basic information about that data set that you could be used to describe that data set. And since you might say that's somewhat of an open question, the question is, what are there specific things that I'm supposed to say? And I like to use this quote from Justice Stewart Potter, who famously described actually pornography, but he says, I should not today attempt further to define the kinds of material I understand to be embraced within that shorthand description, but I know it when I see it. And I want to give you an example of unrich metadata and then rich metadata, and you'll see the difference between them, even though we haven't always defined exactly what rich metadata means, and it might differ a little bit in different contexts. So this is a landing page of a data set that comes out of the Brain Initiative in an archive called DANDY, which is electrophysiology data. And it has its data set ID. They are planning on giving them DOIs, but right now they just have these local identifiers. And this is the title of the data set, 202.006.11 AIBS mouse. And this is the description of the data set that you see. Allen Institute, June 2020, mouse patch-seq ICE-Phys data. So if you were to look at this, you can tell some things about it. You're like, okay, well, the Allen Institute has something to do with it, and it looks like it has patch-seq. But it really doesn't tell you a heck of a lot about the data set. This is an example of unrich data, unrich metadata. Let's contrast that with the data set from Spark, which uses human curators to help people add the appropriate data. So if I look at a Spark data set, this one happens to be on colon, you'll see that the title of that data set is meaningful. It tells you something about the tissue, the type of data that's there, the type of subject they're using. If you look at that and you were searching for data in pig or in colon, a search engine would be able to find that for you, okay? As opposed to that cryptic title that you saw in the previous one. There's a full author list. These are all the people who are responsible for producing this. It gives you information about how to cite that data set. If you're going to use it in another study, it basically says, here are the people, here's a full citation, just like you would cite a paper in an article. It gives you a meaningful description, like an abstract, gives you a link to an experimental protocol and information on how you might use this data, information that you need to know. It tells you what subject and samples are involved. It links to code that you might use in order to use it. Tells you what publication it came from and gives you keywords, grant information, and other things on top of it. So if I were going to look at these two data sets and say which one has rich metadata and which one doesn't, I hope you all pick the door on the left, because it's obvious that this is a much richer description of that data set and what you might need to know to use it than the one on the right. So if you think about these things, right, if you share them with your colleagues, they would be much more likely to understand this than they would be to understand this, right? That's what we mean by rich metadata, right? You're giving a rich context in order to understand when you land on that landing page what this data set is about. So why is it that researchers are really poor about this, right? One of the reasons is that they tend not to think about publishing data sets the same way they would publish a paper. You would never write a title of a paper, 201056 AIBS mouse, right? You would never give that title to a paper. You would give it something meaningful. You would never write the description, this is the ePhys data from the Allen Institute, right? You know that when you're writing papers, you are writing them for a third party that you don't know to understand. So you have to think about when you publish your data in a repository, providing the metadata the same way that you would provide it for a paper, right? You were saying, what is it that somebody else needs to do to understand my data? And the lesson we want to learn here is A, more metadata is always better than less metadata, right? The more data you have about this, the better. But you really need to think about publishing data like you would publish a paper. What information do people need to understand and reuse it? So I wanted to talk a little bit about, since we have some time here, a couple of the other principles. And I'm skipping over accessibility and I'm going to interoperability because I wanted to talk about research resource identifiers. So these interoperability principles are a little cryptic, right? They're cryptic in the sense that you may not know what a formal accessible language is, and in all truth, you don't need to worry about that. Metadata include qualified references, you don't need to worry about that. But this one right here, data use vocabularies that follow FAIR principles. This is actually a very interesting one. And once again, illustrate the power of these unique identifiers, the persistent and unique identifiers. So I thought it would be worthwhile to go through them so you understand why, again, certain journals are asking you for research resource identifiers, for example. So I'm going to give you a little thought experiment here. And this is made up data sets, but I've made up 3 data sets from 3 different laboratories that are studying a rat model of type 1 diabetes. Data set 1 is physiological measurements of kidneys. Data set 2 is gene expression in peripheral organs. And data set 3 is gene expression in major organs. And since I've made these up and I use these examples all the time in different contexts, please forgive me if things are inconsistent because sometimes that happens. But basically, there are 3 different data sets, all of them studying a rat model of type 1 diabetes. So here's my fake data sets, right? And data set 1, there's an age range, there's a name of the organism here, there's a gene. This is where I'm inconsistent because I should have taken that away. And the location where they said they were studying was something called CT kidney. Data set 2, another age range. This one is 10 to 20 days. This is 15 to 30 days. This is name of my organism. Calbindin 2 is the gene symbol and renal cortex. And data set 3 is adult. The organism is something called 6. CR is the gene that they studied, and cortex is where they studied. And so I'd like you to look at these for just a moment and think about what the problems are here if you wanted to combine these data. They're all ostensibly studying the cortex. They're all studying genes. They're all studying organisms that are related to type 1 diabetes. But if I look at this as a human, you see things that there may be things that I don't know, right? I don't know, for example, are they studying the same organism? I have no idea what the number 6 means in this particular context. What would a computer not know? That is, if a computer wanted to combine these data together in some way, it might not know that, in fact, these are all the same gene, just different names for them. It doesn't really know whether this is the cerebral cortex or the renal cortex, and it doesn't necessarily know what CT kidney means, that that's actually the renal cortex of the kidney. You can also see that there are problems with groupings here, but I'm not going to have time really to go into this up here because I do that in another lecture. So the question is, what could you have done differently to make this more understandable, these 3 data sets, to both humans and machines? And again, the answer actually comes down to these persistent identifier, these PIDs. The best practice is really to use identifiers that are issued for these different types of entities to be able to unambiguously identify the same entity across multiple data sets. And these include gene IDs for genes, people IDs, articles, concepts like anatomical structures. All of these have their own identifier systems. I want to spend a little time, because this is the area where dkNET really gets involved, on those identifiers for reagents and tools, the RRIDs, which I mentioned before. So why are these PIDs so powerful? If you actually use these identifiers, you will actually notice that these 3 groups studied the same exact animal. Why do we know that? Because the strain, WFBBRD, blah, 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 is made available through the rat genome database, and they have given this identifier to it, which is what the RRID uses. You will also notice if you go to this record, because it resolves, again, like other resolvable DOIs, that it's got a different name. And some people use this name and some people use this name. You would also know that in this database, they have a hash table in their lab, and they say number 6 actually corresponds to this particular strain. So if the computer looks at the underlying identifier, it really doesn't care the fact that these things are all called different labels. Humans care. But this identifier, in fact, says this is the same rat strain. You'll also see again that there are gene identifiers and that these 3 are all the same gene identifier. When you look down here, you see that cortex kidney or CT kidney and renal cortex are in fact the same structure. But this cortex over here, in fact, refers to the cerebral cortex of the brain. And you'll notice it has a different identifier. So these persistent identifier systems are really powerful in being able to disambiguate things that we look at that on their surface look completely different, or in this case, they look the same, but they're not the same, right? These persistent identifiers are the thing that allows that to happen. So I want to talk a little bit about the research resource identifier, the RRID, because dkNET was instrumental in getting RRIDs accepted as a standard by all the major journals for authors writing their papers, where each resource, antibodies, cell lines, organisms, plasmids, biosamples, tools, is given one of these unique identifiers. And if one supplies this inside of a paper or a database or a lab notebook or what have you, it uniquely identifies that thing and no other thing. So it becomes really easy for a computer to look across papers and say what resources were used in the study, because those PIDs are tied to metadata, and who else has published with this resource without any difficulty whatsoever. So right now, over 1,500 biomedical journals use RRIDs, and all the major publishers support them. So the reason that those are powerful is that, oops, there it is. Once I have that RRID, can gather information together, right? Computers love them, they can search on them, they can resolve them. And that means if papers use them, if databases use them, if you use them in their lab notebook, if your collaborators use them, if other databases use them, you can take all of that information, and for a single resource, you can pull together a lot of that information. That's what dkNET's resource reports do. You can actually go to the dkNET portal and look for RRIDs. Whoops, I don't know why this doesn't advance very well. Okay, oh yeah, here we are. Resource reports, you can find RRIDs, and you can find all of the information that dkNET has aggregated about them. And we'll talk a little bit about that at some future point when we talk about authentication. So the lesson to take from this is that don't gloss over when you see these identifiers. Take advantage of these identifiers, the RRIDs and other IDs, to document your data, to annotate your data. It will be a lot easier for you when you write your papers and submit your data if you have these identifiers than if you have to go and look them up. We find inside of laboratories, we'll look at antibody inventories, and they'll have no identifying information other than this is an anti-actin antibody from Sigma. And you go to Sigma, and there's a whole bunch of them. And they might have a catalog number, but catalog numbers can change, right? And so we really find that getting into the practice of using RRIDs for all the things that you use inside your data is really, really very powerful. Okay, finally, we want to get a little bit to reusable. And if we look at the reusable principle, because ultimately this is why we're doing it. If we didn't care about reusing the data, then the rest of it really doesn't matter. But as I mentioned before, there is a really strong drive for us to be able to reuse public data. The Hypothesis Center is built on public data, and there's very powerful things you can do with it once you can aggregate these things all together. So if you look at reusable, it says that data are richly described with a plurality of accurate and relevant attributes. Again, it doesn't tell you what those are, so people are left to try to figure it out. Data are released with a clear and accessible data license, and data meet domain relevant community standards. So what the heck do these things mean? That's where an organization like dkNET can help you, because it can help you interpret FAIR in the context of your particular domain. So how is it that dkNET does that? Well, first of all, the ones who really enforce standards and the ones that enforce what metadata you should collect about your data are the repositories. You saw how much information Spark wanted from you about your data when you submitted it. So if you know in the beginning where your data are going to end up, it will help you plan your experiments and understand what all that metadata that they're talking about you need to collect, okay? There are groups around the world who have issued these standards, and they will tell you for this type of experiment, you need to know how many replicants you did and blah, 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 right? The rich metadata I talked about was generic metadata about it. But once you get deeply into a type of experiment, there are very specific things that people want to know in order to be able to interpret it. So the lesson here is if you think about where your data are going to end up in the very beginning, it will help you plan what type of data and metadata you need to acquire from the get-go rather than after the fact. And again, comes down to these repositories again. It is the repository that you submit it to that will often be the one that is enforcing this standard. So they will be the ones to tell you what standards are available for your type of data or your domain. And just as a little aside, because biomedicine is very big, if there isn't a domain-specific repository for you or a data type, there's also general purpose repositories available. So you can always find some place to put your data. So to help you, dkNET maintains a list of repositories, as I've mentioned, and they organize it by data type and domain. But we also provide a lot of other information that's available to you, right? We say, here's instructions on how to submit your data. Here's instructions on how to use the data. Does it cost any money? For very, very large data sets, sometimes it costs money. And are there standards that these repositories are going to enforce? So you basically can use dkNET to both find a repository for yourself and then also understand what sort of resources you're going to have to dedicate to preparing your data and what sort of standards you're going to have to follow. Right now, this is just a list that you can search on the dkNET webpage, but we're in the process of developing something called a FAIR data wizard, which will actually walk you through the different stages of FAIR so that it makes it easier for you to achieve that.
That's really going to be fabulous. You just answered my question, which was, can we, how do you know which is the best? So that's going to be fantastic.
So it's going to tell you about it. And as I mentioned, we do have what standards these databases require, and there are also other sites like Fair Sharing, which will tell you things like metadata standards. So for example, if you're going to put your data into Geo or Array Express, which is microarray data, it says, well, you're going to need to use the Miami standard for microarray. Then you can look at the microarray and it will tell you, these are the things that we're expecting from you. This is the type of data we're expecting from you. If you talk to repository owners, the chief reason why people come to and leave their repositories and leave fields blank is they don't have the information at hand. If you have the information at hand, then it is very easy to deposit it, okay? So it's really important to sort of think this through from beginning to end. And finally, again, we all need help with this. We need to understand this. And I've mentioned the fact that NIH is actually going to expect you to put a plan in on how you're going to do this. And there's actually been a requirement for a while, if you've submitted a grant lately, for something called a data management plan. They've now made that more specific with a data management and sharing plan. But there are a lot of excellent resources that are available to you to help you understand specifically what you need to collect. I went through it at a very high level, but there are a lot of resources, a lot of them coming out of the library, that will help you prepare your data management plan. And then you can use additional tools like dkNET to do your data management and sharing plan. Again, dkNET is developing these things into a wizard, but right now, at the very least, if you go to dkNET, we do have links to all of these. So here's Data Management 101. Here's an entire course on how you can manage digital data in your research lab. And there's a tool that actually lets you build a data management plan. So there's a lot of help out there. You're not alone. So dkNET, I hope you can see, is a resource that can be used to help you navigate this new world of data sharing that's coming out. We're constantly updating our materials. We're constantly trying to make them better. We would love for you to be able to visit our site and take a look at the information for best practices of managing research data, our list of approved repository. And if there is no repository there, or you're confused, we have a form you can fill out to request us to take a look at it to try to help you with this. It's a constantly changing field. And as I mentioned, coming soon, the FAIR Data Wizard, which will be an automatic tool which will help you with this. So I know people are probably feeling overwhelmed with this. So once again, I'll just repeat, if you have questions, oops, you need to ask dkNET. Okay. If you're a student, please contact us. If you're a student, please apply for dkNET Summer of Data program, where we actually go through these types of materials in more detail. And also ask your library, because there's a lot of expertise out there for people who are interested in this. And I think I will end it right there.
Fantastic. I think this cleared up a lot of sort of black holes that are out there for people. And I thought it was a really lively way to talk about some kind of very granular amount of data that's out there. So it was great. I think do you have any timeline for the Fair Data Wizard?
So we have a proof of principle right now. And so I imagine it will be probably in the fall is when we'll release the version.
And what kind of onboarding do you generally see for new researchers coming to start to interface with dkNET, learning to use your tools, and how long does that usually take someone to get up to speed on sort of this whole fair data runway?
So we deliberately made those pages fairly easy for people to read to get started, and then with links to appropriate tools. So I think trying to grasp the issues is fairly easy. In-depth on FAIR is you have to be a little bit more dedicated, because you can see there's a lot of complexities to FAIR, but we tried to break it down into the simplest steps that you really need to pay attention to. Using the repository list, we've had very good feedback on it. Even though it's not a tool, it at least puts all the information in one place so that people can do it. Actually, the NIH data science team referred to it in some of their presentations. Are like, see, this is a very easy way to actually look at this, and we're constantly improving them. So I wouldn't say that. I think kind of getting started is easy. Trying to figure out how you're going to handle this in your laboratory is, of course, a much bigger problem. It requires knowledge, it requires tools, it requires planning. And that's the big change that we're going to start seeing, I think, in laboratories as a result of NIH's policy, and also in institutions. They know that they're going to have to support this better. And they've been building bits and pieces of this over the last few decades. They haven't had a lot of uptake necessarily or interest from the researchers. The mandate is now going to close that gap, because the researchers have to use it. The good news is we would not have this policy if everybody had to start from scratch. There's tremendous amount of expertise out there. dkNET is one place that really is kind of claiming the space of interpreting FAIR for DK fields. But there are other organizations that are doing it for neuroscience. There are scientific societies that are saying, okay, right, this mandate's coming, we need to pay attention to this. So what are we going to do? I think in the beginning, everybody is going to go in fits and starts, because anytime you have something new like this, it's going to take a while. NIH, I think, was actually criticized by some, especially the open science, right? The ones that are really robust, saying it should have been stronger, it should have been stronger. But I actually respect the way that NIH did this. Biomedicine is so big and vast that trying to create like these really, really harsh mandates for fields that maybe have no infrastructure is going to be very different than if you're in genomics, for example, where there's a whole bunch of infrastructure. So there has to be flexibility.
I totally agree with that. It's going to be hard getting everybody on the bus. Are you seeing, but I think it's an absolutely valuable program, and it's really going to, like you said, this is so cool to be part of really the changing the way that science does business. And this is an inflection point. It's really going to change things. So it's kind of exciting in that way.
That's what I think.
Yeah, I do. I mean, it really is. I mean, it's daunting. It's like, oh no, one more thing I have to learn. But are you seeing, this is sort of like one of my last questions. I know you guys have a hard stop, but what do you, how do you see the people that are sort of adopting this pretty, quickly or already are on board? Are they designating someone like a lab manager to sort of like pull through the whole team? Or are they really saying, hey, everybody has to train up on this? Or what's the real life picture look like?
So I think that's interesting. I think for the most part, people right now are being left on their own. But I think one of the reasons that investigators are happy to have their students go to the Summer of Data is that they can bring these approaches and tools and techniques back to the lab. Most PIs, of course, aren't going to be managing their own data. They leave it to everybody to do that, but they're generally the ones that are hurt by bad data management practices, because then they don't have control of data in their own laboratory.
Right.
So we are seeing at the National Academy workshop I mentioned, there were some researchers there, some fairly high-level researchers who are extolling the virtues of formal data management in their lab, because they either work with people who could help them to set up a system in their lab. And they really said, once that was done and once people got used to working through that system, the benefits were so immediate and tangible that there was no resistance anymore as to why one would want to do this. There were some really good examples of that there. The best example was exactly the one I used. They had submitted a paper to a journal. The journal said, I'm sorry, now we require all the data that underlies a figure to be submitted separately, right? So you can reproduce each figure. And I've seen other groups, myself included, because we don't always necessarily practice what we preach, scramble then to say, oh, wait, here's my folder. I thought I understood what I did in here, but I don't understand it at all. And he described, I pressed a button, here was all the data. I bundled it up, I put a DOI in it, I submitted it to the journal, thereby saving hours and hours and hours. And sometimes, again, you can't even reproduce it because you yourself don't remember what it is that you've done or where you put things. So I think that the hope here was that, again, that people would have a sense of excitement about this, and that NIH does say, right, you can put the cost for what it's going to take you, right, to do this properly into your grant. We don't expect this to be layered on top of it. You're going to have to put money in your grant. That's why the data management plan is so important, because it helps you think about, well, what am I going to need? Will there be a cost for it? If I'm going to be saving and sharing spreadsheets, probably not, right? That's not going to cost you anything. If I'm taking terabytes of data, you don't walk around after you've got the terabytes and say, okay, now what do I do with this? You need to plan for that, and you need to allocate the resources that are going to be necessary for every step of the way.
Yeah,
so
that it will be built in. So it's not like, you have to sort of like draw from other resources. You build that into the grant and then you deliver against it.
Exactly.
Well, we look forward to the next one. Just sort of going through these, as we've called them before, the treasure trove of tools and resources that are found at dkNET. And I think as we parse this out to the community, they're going to see more and more reasons why they're going to watch this presentation in total, because it's interesting, and it's cutting edge, and there's a lot to learn. And frankly, you got to learn it. So
that's what I said. It's no longer a matter of like, should I or will I? It's like, nope, you have to.
Thank you again to the dkNET team. You've been incredible, and we look forward to our next discussion with you.
Thank you for inviting us.
All right, we'll talk again soon. Thanks again.
Bye.
Bye-bye.